Visible light has wavelengths between approximately 400 and 700 nanometer. Light first enters the eye through the cornea, which is the transparent, curved outer layer that constitutes about 1/5 of the sclera, the white outer layer of the eye. The cornea acts like a camera lens, refracting light to initiate focusing. After passing through the cornea, light moves through the pupil, the black circular opening in the center of the iris, the colored part of the eye. The iris adjusts the size of the pupil, controlling the amount of light entering the eye, similar to the aperture of a camera. The light then passes through the lens, which is flexible and changes shape, thanks to the ciliary muscles, to fine-tune focus. The lens further bends the light rays, projecting an inverted image onto the retina at the back of the eye. The retina is a light-sensitive layer of tissue containing photoreceptor cells rods, which are responsible for vision in low light and do not detect color, and cones, which are responsible for color vision and are concentrated in the fovea for sharp detail. These photoreceptors convert incoming light into graded potentials. The graded potentials are relayed to bipolar cells, which, when sufficiently stimulated, generate action potentials in ganglion cells. The axons of ganglion cells form the optic nerve, transmitting these action potentials to the brain for processing. Approximately 90% of the visual signals transmitted by the optic nerve are directed to the lateral geniculate nucleus for conscious processing, while about 10% travel to the midbrain's superior colliculus for reflexive responses, such as meiosis for pupil constriction and mydriasis for pupil dilation. From the lateral geniculate nucleus, visual signals are relayed to Brodmann area 17, the primary visual cortex, and subsequently to Brodmann area 19, the secondary visual cortex. Some pathways also lead to the medial rectus muscle, which is controlled by neurons in the oculomotor nucleus and is responsible for the reflex of convergence, allowing both eyes to align toward a near object. In contrast, the ciliary muscle and sphincter pupillae play essential roles in accommodation. The ciliary muscle adjusts the lens shape for focusing on nearby objects, while the sphincter pupillae constricts the pupil, enhancing depth of field and reducing light entry. It is important to note that our vision is not complete. The brain fills in extra spaces and integrates information from both eyes to create a three-dimensional, cohesive image, enabling us to perceive depth, movement, color, and fine details.